Hi, everyone. Welcome to Physics 380, Molecular and Cellular Biophysics. My name is Dr. Brenda Lee, and I'll be your instructor for the term. In this lecture, we're simply going through the course overview, as well as an introduction to molecular and cellular biophysics. So in this first lecture, I want to go through a quick recap of what we learned from previous courses, just so that we know what kind of information and knowledge we need to be able to understand the knowledge that's being presented in this course. Afterwards, I'm just going to quickly go through all the course details, as well as who your teaching team is, what's the schedule of topics, and what are the key deliverables for the course so that you can get your grades. After that, we'll go through a quick introduction as to what is molecular and cellular biophysics, and then go through a brief summary of what we've learned in this lecture. So what should you already know about? Well, we should have some calculus background so that we can do the mathematical modeling required for the concepts being taught in this course. You should have Physics 112 or 122 already, which means you should have a strong background in general physics. You should also have experience in chemistry, specifically Chem 123. We will be looking at chemical reactions and some biochemistry for this course. So in terms of Math 127 and 128, that's the calculus that is highly recommended for this course because we are going to talk about algebraic, logarithmic, and exponential functions as part of the models that we create to model our different macromolecules. Definitely know some statistics for chance and probability when we're discussing that, as well as know some differentiation and integration for when we do have to present mathematical models. Next, from general physics, the two key topics that we are going to discuss are going to be simple harmonic motion, as well as energy and entropy. Some additional topics would be electrostatics and magnetism. Chem 123 was chemical reactions. We are going to be focusing a little bit on the ideal gas law, as well as rate equations for chemical kinetics. These are biochemistry topics. I will be presenting them again briefly at the beginning of each lecture. I ask that if you are unfamiliar with some of these topics, feel free to reach out because there are a variety of students taking this course, some with stronger physics background, some with stronger chemistry background, and some with a stronger biology background. So if you're struggling with any component, feel free to reach out so that I can supplement additional information for you. Okay, so now on to the course itself. The important stuff is who, as well as the outline, the grading distributions, as well as the types of assignments and projects that are going to take place within this course. So how is Physics 380 run? This is a remote course. So there are two types of lectures. There's going to be core lectures that are delivered through pre-recorded videos like this one right here. Basically, I'm going to be posting the slides on Learn. You can download the slides ahead of time as PDF files, and then you can watch the pre-recorded videos, which deliver the core content of the course. So use this as the textbook so that you can study from. There is going to be information that I go through verbally that is not included in the text documents for the PowerPoint slides. So make sure you have those ready so that you can take notes as I go through the pre-recorded videos. I also have live lectures that are going to take place each week and are recorded during regular lecture slots. There is going to be a poll that I'll be releasing so that everybody can vote on the best times possible to hold this lecture. During this lecture, I will be reinforcing content from these slides, as well as introducing new topics and new examples to offer a chance for us to engage in discussions about biophysical modeling. So I guess now it's time to introduce myself. My name is Brenda Lee, and I used to be a student here at the University of Waterloo from bachelor's to PhD, and now as a faculty member. So I've gone through a lot of different experiences as a student and as a grad student. So all of this experience does help me in teaching this course because I used to take this course as a student with Dr. Bae-Yun Ha. And then later on as a grad student, I did a lot of work in molecular biophysics. In terms of office hours, I do have office hours through Microsoft Teams or email. You can contact me through Microsoft Teams anytime. We do have a custom Discord server that I've set up so that everybody can connect with each other and get to know each other a little bit better, and allow you a chance to hop to different channels to chat about different things, whether it's something fun or whether it's something related to the course. You are also allowed to collaborate with each other on certain assignments, as well as the project at the end of the course. Our teaching assistant is Olya Changizi. Their email is given here. You can reach them through Microsoft Teams or email as well, and they will also be on the Discord server providing support. Now, the course topics and schedule, I'll leave you to read all of this, but essentially we are going to be starting off with an introduction. We're going to discuss some fundamental theorems for biophysical modeling at the molecular level, as well as different types of intermolecular interactions. Once we have that knowledge, we can apply it to different types of macromolecules, from proteins to DNA or nucleic acids to lipids. And so we are going to focus more on the molecular side of this course. So your final grade is going to be calculated using this scheme over here. There's only one scheme, so there's going to be assignments worth 25%. There's approximately 7 to 8 assignments throughout the term. I definitely do not want to give you more than one key deliverable per week. So I'm hoping to keep things simple that way. There will be a literature review project at the end of the term that's due, and that is going to take up 30% of your grade. So for the majority of the term, please do keep the literature review in mind as you progress throughout each week. There will be milestones and checkpoints to make sure that you're keeping on track with everything as well. There is a midterm and a final assessment. Midterm is worth 20%, cumulative. Final assessment is also cumulative and worth 25%. They are both essentially take-home exams. There will be a bonus worth 3% that will ask you to try and enhance or improve one of the models that I present throughout the course for one of these macromolecules. So in terms of weekly assignments, there's a total of 7 to 8 assignments, and the lowest grade will not be dropped. So make sure you do complete each of the assignments and do seek help if you are struggling. We're going to use CrowdMark to submit assignments and to grade assignments. If you don't have a scanner, there's a good app called Cam Scanner that you can use. That has worked well for many of my students in the past. Another thing to note is that it's important to watch lectures. We are going to go through practice problems together, specifically in the live lectures, and the solutions to the assignments will be posted after their due dates. Now for the term project, the most important and the biggest key deliverable for the term. It is a literature review, and one of the things that I would have really liked as an undergrad student was more experience in writing and doing research and gathering information so that I could write a good report. So this course really does try to enhance that experience for everybody. I'll try to guide you through the entire process of writing a literature review, from doing a search for different references and journal articles to being able to compile and condense all the information that you receive from all these articles, as well as summarizing everything so that you can organize it properly when you present your literature review. So these literature reviews are really important when you're doing honors projects in your fourth year, for example, as well as graduate studies. It's good to understand what they are now so that you're prepared when you have to do them in the future. Literature reviews usually range from 5 to 50 plus pages, depending on the topic you've chosen and the scope of your project. But of course, we can't have like a 50-page lit review for this course, so we're limiting it to 10 to 15 pages. That means the selection of your topic has to be carefully done. You don't want a topic that's so broad that you end up not having any focus whatsoever. You want to have a topic that is specific enough for you to have many resources to look at and allow you to summarize things in a manner that you can explain that topic in a very detailed manner. So there will be a lot of guidance provided, as well as milestones throughout the term that will help you stay on track. So the first milestone is going to be picking your topic, because that's the most important thing, and sometimes it does take weeks to finalize a topic. We're going to be using the Dropbox on Learn to submit your topic. It's worth 2 marks, so that there is grades for being able to complete things on time. Milestone number 2, you have to create an outline. So it's worth 3% or 3 marks. We're going to use Dropbox on Learn again. And it should include the following. When you've chosen your topic, you should be able to do some research, read some papers related to your topic to try and fine-tune the types of studies, papers, and conclusions that you've read throughout all of those papers. So I'll be asking everybody to have at least 10 plus references already read and summarized, and you can simply create a approximately 300 word description of your topic and to explain what you are trying to accomplish in your literature review. Now, keep in mind, the 3% is a completion grade, and after you create your outline, the teaching team, myself and your TA, will be going into each of the outlines to provide feedback. That way you can continuously improve throughout the entire term. So essentially, you want to create a table of contents after you've read your 10 plus references, and then be able to summarize and organize your thoughts so that it makes for a good story to tell for your literature review. For milestones number 3 and 4, they're basically the same one. There's going to be 2 checkpoints throughout the term after you've submitted your table of contents to allow the teaching team to review the draft of your literature review. You only have to submit at one of the checkpoints, but there are 2 checkpoints in case you want to receive feedback twice. So if you submit at the first checkpoint, you're going to be graded out of 15 marks, which is worth 15% of your project. Now, with these 15 marks, it is going to be graded based on the quality of the work that you've done. So if you submit at the first checkpoint, and let's say you get 8 out of 15, then you can resubmit at your second checkpoint to get the 15 out of 15 after we've provided you with feedback from the first submission. If you choose not to submit something in the first checkpoint, then you will not have a chance to get feedback by the second checkpoint, which means the grade you get out of 15 in your second checkpoint will be your final grade for that third and fourth milestone. But it doesn't mean you won't get feedback. You will still get feedback after the second checkpoint, but you would have only one chance to score out of 15. Now, the last milestone, milestone 5 and milestone 6. Milestone 5 is actually a plagiarism check. There's going to be a Dropbox folder on Learn for you to submit as many times as you want, and it's simply to check for plagiarism, because you do not want to have a Policy 71 violation. Submit your paper to the plagiarism checker for Turnitin to see if it's okay. You can always ask me for support once you get a percentage of similarity. Sometimes you have 5%, some people have 30%, depending on how big your reference list is and what kind of paper you have. That value is sometimes accurate and sometimes inaccurate. So just make sure that you are submitting something that is original and not plagiarized. And after you've confirmed that in milestone number 5, you can go ahead and submit your final project in milestone number 6. So that final project and the final submission is worth 100 marks and worth 80% of your final project grade. So the rubric is going to be posted on Learn so that you know how I'm going to be marking the final lit review project. And so do take a look at that before you submit the final draft, just so that you can make sure that everything that I'm looking for is included. Now, because the final version is due by the last day of classes, there will be no late submissions accepted. So anything that is submitted past the last day of classes will get a 0%, unfortunately, because it is going to take time for me to read through many papers. So I can't accept anything past that date. So for the literature review, what do you have to include? The completed literature review needs to be uploaded through Learn's Dropbox. The condensed lit review, and I call it condensed because usually lit reviews are like 20 to 50 pages. It should consist of a title page, an abstract of no more than 300 words, the main component of the paper, which needs to be 10 to 15 pages in length without pictures or reference, and it has to be single-spaced in letter-size paper. So citations should be numerical and in order of appearance, so try using ones or footnotes to help with that. You also must use subheadings to help organize your paper. Outside of that, we have references. So at the end of your project, you should have at least 20 references published within the past 5 years for consistency and to make sure that the information that you're presenting is the most recent and up-to-date information from the literature. The other thing I want to stress is to not use websites for the literature review. As you work on research projects and as you move on to graduate studies, if that's your interest, then you are going to have to find sources that are primary sources, either textbooks, journal articles, or manuscripts. So apart from the literature review project, there's also a bonus project. It's worth up to 3% on top of your final grade. Basically, throughout the term, I'll be presenting simple models for macromolecules, whether it's trying to model the electrostatics of DNA or something similar. So for all of these models, these are simple ones, and it allows students to learn about how models are applied to real life. For a bonus, I would like to ask you to try and think about, design, and implement an improved model for any of the models that are presented throughout the course. So what do you want to submit, or what do you have to submit? You have to submit either a written document, a presentation, or a program script if you want to use programming language to try and create this model. But you do have to provide some way of communicating how you would like to improve the model and how you've accomplished this. And so full details are going to be on Learn under a bonus folder. Please check that out if you're interested. And the deadline would be the last day of classes. Now for course materials, first of all, there are the lecture notes that I post as my slideshows, as well as the videos that are published on Learn. Please note that the lecture notes include figures taken from external resources, and these lecture notes that are mine cannot be republished or handed out by students under any circumstances, or else there will be a violation of copyright. So please try to keep everything confidential. Textbooks, there are none required for this course, but there are many resources. There are so many different types of textbooks for biophysics, and each one is catered to a different audience. So although textbooks are not required, they offer additional resources if you desire to explore more topics within molecular and cellular biophysics, and if you want to know more detailed information that the lecture notes do not provide. So these are the 4 different textbooks that I would recommend. There's Physical Biology of the Cell, which gives you a very basic background into all the different physical theories that are required to model different types of macromolecules. Essential Cell Biology is purely a biology textbook. It goes through all the different types of molecular processes. That is very handy when it comes to trying to determine the parameters to model something. Molecular Driving Forces is another good textbook. It basically focuses on the different forces that are involved and the mechanics involved in different molecules. Then you have Biological Physics from Philip Nelson. This one's pretty good as well. It goes through all of the physical foundations for different real-life applications of biophysics. So midterms and final exams, I want to stress that the midterm and final exam is just a take-home assessment. We don't have an official midterm or final exam during any official period. So I want to wrap up all of this by the end of the term or the end of classes. The midterm assessment will take place in week 7. It's a take-home assessment. You have 24 hours to complete it. There's going to be an individual component and a group component, and you can submit the assignment through Learn. Literally the same thing for the final exam. It's in week 12, the week before the last day of classes. It's a take-home assessment. It's going to be in groups. There is going to be an individual component as well. Once again, you have 24 hours to start and submit your final exam. More details are going to be coming after the midterm. Some additional administrative stuff. If you miss a test or an assignment, you have to submit a VIF or a verification of illness in order for me to do something about it. Please note, you should not plagiarize because it's going to lead to a case of academic offense, Policy 71. Always hand in individual work unless otherwise specified. Now, for viewing assignments and exams, I want to say that we are going to try our best to hand back assignments within a week, but let's just say assignments will be handed back within 1 to 2 weeks, and that exams will be available for viewing within approximately 1 week, with the teaching team working to mark the exam together, including myself. So is Physics 380 useful? Well, Physics 380, I think, is very useful. It's, first of all, an extension of Physics or Bio 280, an introduction to biophysics, but it focuses on something completely different. Whereas Physics 280 focuses on the macro scale component, so basically looking at what takes place in the human body, we are taking a look at what takes place on the molecular level. There is some overlap with Physics 225, Modeling Life Physics, where that course presents a lot more statistical analysis of different models. We also know that this course can be very useful for upper year courses such as Physics 395 and 396, the biophysics of therapeutic methods or the biophysics of imaging. So let's go through an introduction to cellular and molecular biophysics now that we've gone past all the administrative components of the course. We're going to cover some definitions, and we'll focus on the molecular level, and will also take place at some of the examples of interactions and structures that we'll be talking about in this course. So first of all, let's talk about subsets of biophysics. Let's define biophysics first. It's going to be defined as the bridge between biology and physics. It's what attracted me to this field in the first place. Biology usually presents a problem, and physics usually offers us tools to solve that problem. So it gives us the quantitative, abstract, and predictive knowledge and foundational skills to be able to break apart a biology problem that's usually very complex, simplify it into a model, and then predict an outcome. So with molecular biophysics, which is a subset of biophysics, we're going to be taking a look at biological problems on the molecular scale. So that means we're looking at biomolecules and interactions with other molecules. For example, how do proteins self-assemble? How do they interact with each other? How do proteins interact with other types of molecules? Those are molecular problems that we can model using our physics knowledge. With cellular biophysics, this is a subset of biophysics focusing on physical principles used in cell function. So this isn't too much of a focus in this course. We'll be discussing this a little bit at the end of the course, but it won't be a huge focus. The main focus is on molecular biophysics. And just as an example, here is a kinesin molecule it's a kinesin transporting a vesicle using protein dynamics on the nanoscale. So it's pretty cute. It looks like it's walking, but there are different forces and different concepts behind all of these actions taking place. There's attachment taking place at the head of the kinesin, and there's going to be a releasing and attracting force and a pushing and pulling force provided by the walk that's being created by this molecule. So how does physics play a role in biology? Let's start off by mentioning that when you want quantitative data, you need a quantitative model. So before you even get quantitative data, you need to be able to prove that your model compares well with a testable prediction of biological phenomena. So basically, we have to make sure we have a good model before we even make a prediction. So one of the first steps of building a model is determining which features of the problem are of central importance, basically the variables and or parameters, and how they're related to one another. So physicists were one of the first ones who invented ways of approaching biological problems that could be very simply solved with a microscope or visual observations. A physicist tends to bridge the worlds of theory and experiment, and that's where biophysics was born. So nowadays, this has proven to be very useful because we can use physics to model and analyze complex systems, or even manipulate single molecules. So biophysics is still considered a budding field with lots of researchers gaining a lot of traction, with lots of questions to answer and many discoveries to be made still. This picture, though, is an example of the proof of DNA as a double helix. Without physics knowledge and background, this X-ray diffraction image would have not been possible at all. So it was actually a PhD student who took this, Raymond Gosling. Unfortunately, he gave the image to his then supervisor, Rosalind Franklin, who showed Maurice Wilkins, who then showed James Watson, recognizing it as a helix and putting 2 and 2 together with Francis Crick to discover the structure of DNA. So that is the story behind how this image came to be. It was a PhD student who was a physicist. There's actually a famous quote from Francis Crick in his book, trying to summarize his goals and what others should attempt to do. He said that almost all aspects of life are engineered at the molecular level, meaning all biological function. He also said that all approaches at a higher level are suspect until confirmed at the molecular level. This means that we have to understand how and why something works at the smallest level with great detail before we can assume the cause of any higher functions. So that is the core purpose of molecular biophysics. We want to be able to explain how something works at the smallest level of detail so that we can start to make predictions about how it can cause higher function. So I know this course is called Molecular and Cellular Biophysics, so we should be able to try and differentiate the two of them. First of all, I have mentioned that this course focuses a lot more on molecular versus cellular, and that is true. So starting from the top, we have cellular communities, which is from big to small. The biggest would be cellular communities. As we break it down, a cellular community can be broken down into individual cells. Now, in order to make up an individual cell, you have to have different molecular structures. So we break it down even further into this. And what makes up molecular structures? It's biomolecules. So then we have to break that further down into biomolecules. From there, we have simple molecules and then atoms. The majority of this course will start from biomolecules and then slowly discuss how biomolecules interact with each other to form molecular structures. So that's essentially the whole focus of this course. In this course, life takes on a different meaning, especially in biophysics. So the pursuit of understanding the nature of life means that we have to investigate the properties of living organisms and their constituent materials at different levels. We all know that living things are made of cells. And in fact, we know that organisms are constructed from the 4 great macromolecules: proteins, nucleic acids, carbohydrates, and lipids. All 4 of these are pretty different, and we call this diversity in life. However, despite this diversity, there's also similarities when you break things down to their most basic fundamental components. So these diverse macromolecules all share a structure that's based on carbon, oxygen, nitrogen, hydrogen, and some sulfur and phosphorus. So it's mainly the way in which this set of atoms are connected to each other that creates the special properties of each of these macromolecules that are found in living systems. So to me, that's pretty cool. And because of this, we call this the molecular scale and also unity in life. So understanding what unity means and diversity in the definition of life in biophysics, that's important. Even with my research, for example, if a drug has 10 or 12 amino acids and doesn't work, you could potentially change one of those amino acids and it completely changes the functionality of that drug. So throughout this course, we're going to be taking a closer look at the unity of these macromolecules at the molecular level and then build up towards diversity. So moving forward, cells use the same chemical building blocks to build their biomolecules, such as DNA, protein, etc. And we're going to see how those building blocks interact with each other to give function to each macromolecule. Since we're looking at the molecular level, we can also assume that their physical and mechanical properties are very similar. So that allows us to use the same physical models to represent their properties. For example, electrostatics and different interactions between them. So here is a picture of the 4 great macromolecules and a label for each of them. So over here, we have nucleic acid, basically DNA or RNA. It plays a critical role in storing and transmitting genetic information. This slide explains the functionality of each of these macromolecules. It's very important to remember this. Over here, you have a tiny little lipid or phospholipid. It stores long-term energy, protects us through the formation of barriers and membranes. Over here in the middle, you have a large globular protein, or in other words, just a protein. They do most of the work in cells and help with the structure, function, and regulation of the host. Over here, you have different elongated structures, which can represent carbohydrates. These are used for energy storage, in other words, quick energy storage versus long-term energy storage, as well as surface properties. So in a lot of cases, you could have proteins mixed with lipids or carbohydrates based on their functionality and what they're presenting on the surface. Now, you're all probably familiar with the central dogma of molecular biology, or maybe you've heard it before. This describes the flow of genetic information within a biological system. So it takes you from DNA replication to transcription to translation. So basically, how do we read DNA so that we can create proteins? That is essentially the central dogma of molecular biology. But the key thing here isn't the process itself. It's the fact that differences or different arrangements of building blocks can create different pieces of information. So maybe a certain piece over here creates a particular protein, so on and so forth, or a different part of the protein. So when we look at a model, we have to make sure we look at the building blocks and links within a chain and see what kind of information they carry in order to see the entire picture. And this is something that we'll discuss as we move on throughout the lectures. For now, let's just say the central dogma of molecular biology is going to describe how we get from genetic information to a protein that carries out the function written by our genetics. So just to wrap up this lecture, I'm going to present a few examples of different types of models that we'll be covering throughout the course. First, we will be talking about modeling DNA. So we can model only one aspect at a time. DNA is something we'll cover more towards the end of the course because it is a little bit more complicated. We're going to start off with proteins. But over here, we can model DNA in several different ways. We can model it by sequence, for example, basically a string of letters. We can model it by binding site. So where does it bind? We can model it by a charged rod. So that would be introducing electrostatics. We have an elastic rod, in which case we're introducing spring constant and trying to look at curvature, so on and so forth. Then we can also model it by random walk, in which case we usually write a program or we use some sort of computational method to figure out how it folds or is arranged in free space. Now, when we talk about modeling proteins, it's a little bit of a different approach. Proteins actually have so many different configurations. We usually need separate models for each type of structure. There's primary, secondary, and tertiary, and quaternary structures. So when we're taking a look at each of these individual structures, which symbolizes how much folding has already taken place, we're going to be looking at hydrophobic, hydrophilic amino acid interactions. We're going to be looking at random walks, basically computational studies. We're going to be looking at alpha helices and beta helices for secondary structures, and how those two different types of structures give rise to different types of proteins. So once again, many different ways of modeling things. I listed a few over here. The random walk model can be done computationally or graphically. Because computational physics is not required for this course, we are going to focus on how to represent this graphically, while also presenting some additional bonus information for anyone who's interested. Next, we have lipids. So we will be looking at modeling lipid membranes. These are modeled as different types of surfaces based on phospholipid properties. So phospholipids actually have a hydrophilic head and a hydrophobic tail. So the head and the tail means that you can have different configurations of a lipid. You can have different sizes and shapes of those hydrophobic heads. You can have different lengths and kinks, kinks as in how much they get farther apart, to the hydrophobic tails. Because of this, it means that we can have various different types of surface properties. If there's more kinks, then the membranes are a little bit more spaced out. If they're more straight, then the membrane is more tightly packed. If you have a larger head, then you have different types of properties at the top and more surface area for things to attach to. So in terms of this, we have a lipid membrane over here, and it can be modeled by an array of springs. We could assume that each of these is a spring. We could model it as a random surface, could model it as an RC circuit, in case you have ion channels and you want to see if there's any resistance going through, or if you want to model the membrane potential at the top and bottom. You can also model it as a semi-permeable barrier to see how much or what kinds of ions and molecules can pass through the membrane based on how spaced out it is and surface properties. So we'll be going through that quite a bit at the very end of the course. Now, with modeling cell movement, this is something we'll talk about as well. When we talk about a cell, we can model its movement in several different ways. We can model it as an array of receptors for a ligand of interest. We can model it as a swimmer with a rotating tail. We can model it as random walk if there's another term for this, it's Brownian motion. So basically the cell is moving in a random fashion. We can also model it through a genetic network. So these are things that I will explain later on as an extension to the course. Our main focus for the course is lipids, DNA, and proteins. Everything else will be wrapped up at the end. Something else that we can model is solutions. There are many types of solutions. This kind of treads more into biochemistry, but we will be taking a look, if we have time, about how solutions can be idealized as lattices, mean flow, a viscous medium, and more. So for example, you can have over here, you can have a lattice solution where you have a lattice of discrete positions where solutes might exist. Over here, you have modeling as a fluid with some sort of flow. So you can calculate the average flow, things like that. You can model solutions as a viscous medium that exerts a drag force on objects that go through it. And this can be tied back to physics 280 as well when we're talking about blood flow, which is described as a viscous medium. Over here, we have a diffusive landscape, so basically a fluctuating environment that gives rise to random motion in macromolecules. You have a hydrophilic medium that readily dissolves polar molecules but repels oil-like molecules. And finally, you can have a dielectric medium, which is based on electrochemical studies. So for us, we'll be focusing on modeling solutions as a hydrophilic and dielectric medium because our macromolecules are going to be in fluid all the time, and being able to understand the interactions within a solution is fairly important because that is where our macromolecules exist. Finally, the question is, can everything be a spring? In physics, many things can be modeled as springs, and there are a lot of biological models that are based on simple springs in classical mechanics. So from here, the assumption is that when a spring is relaxed and at resting length, the energy involved is at a minimum. So when you're looking at biological models, for example, if you're doing some atomic force microscopy imaging and the tip of the cantilever bends, then that means greater energy is involved. If you have, let's say, a protein or something with one side attached to the bottom of whatever you're attaching it to, and another side to the chain attached to the top of whatever it is, if you stretch it, then more energy is involved. If you relax it at its state of equilibrium, then there's no energy involved. So things like that, we can model different macromolecules as springs to help us define it mathematically and physically. So now on to the summary. So summary of this topic, we took a look at the significance of molecular biophysics. So we presented some physical tools that allows us to study life on a molecular level. We have gone through a few different types of slides about the complexity of modeling biophysics and different biological systems. So the key takeaway message is that there are many different ways to take a look at one biomolecule, but we have to pick one way of looking at it and hope to get some sort of information from it. Molecular biophysics is primarily based on the idealization of a biological system into a simplified model. That makes sense because as you include more parameters, your whatever function you create or whatever model you create is simply going to become more complex. So complex that it'll be harder to predict. So it is important to be able to simplify our models so that it's easy to make predictions from and to get a deeper understanding of how core and fundamental interactions take place. So every model that we choose, no matter which one, and every property that we pick can provide insight into how things work at the molecular level and why higher levels behave a certain way. So the goal for this course is to be able to model things on a fundamental level, understand the fundamental principles behind different interactions, and from that be able to derive or predict certain behaviors such as protein folding or lipid self-assembly.